Hi, and thank you for watching. This is my first recorded lecture. It's on the cardiovascular system, and I am only using the audio component. There is no video component. So in the lower right corner of your screen, you may see a gray square. Ordinarily, my face speaking would appear there, but you know what I look like. We don't need the video component. On later slides, I'll be able to draw on the image, and that should come through in this video. So we're covering the cardiovascular system in 3 parts. In part 1, we'll cover the heart. In part 2, we'll focus on blood vessels. And in part 3, we'll focus on blood. Of course, everything is interconnected. So at various points in time, we will be talking about the heart, blood vessels, and blood simultaneously. In our last face-to-face class meeting, we started this material. In part 1, we got up to approximately slide 105. I'm going to start from the beginning here. In part 1, we'll start off with an introduction to principles of exchange and circulation. We'll look at some organisms that don't have cardiovascular systems and then why other organisms do need cardiovascular systems. And then we'll revisit body fluid compartments. We've previously talked about body fluid compartments way back in the beginning of the semester. And we need to revisit them because exchange between body cells and the environment are mediated by the fluids that are in these body fluid compartments. And then starting with point 3, we will focus thereafter specifically on the heart. Okay, so I hope you remember that every single living cell across all organisms, every living cell needs to exchange materials with its environment. Living cells need to bring in nutrients that act as building blocks so that they're able to synthesize their own molecules. All living things need to exchange gases, and all living things generate waste products that need to be eliminated from the cell. So every living thing must exchange material with its environment, and natural selection arrived at 2 different solutions to the problem. So shown here, we have just a single cell, a unicellular eukaryote, and that cute little puppy is much more like you. It's a multicellular creature. Right. So one solution to this problem, the problem being that all cells need to exchange materials with its environment, is that the organism's body plan is either unicellular or the multicellular organism is thin and flat, so that all cells in that organism's body are in direct contact with the environment or not too far away, maybe 3 or 4 or 5 cells thick. And so these are examples of creatures where they consist of either just a single cell, like that paramecium up top, or they have a body plan, a multicellular body plan, but no cell in that organism's body is particularly far away from the environment. So in that moss and in that flatworm, the deepest cells in the bodies of those organisms are perhaps 4, 5, 6 cells away from the environment. And that's enough for gases to diffuse, for waste products to diffuse through the organism's body and into the environment. But what about large, more complex organisms like that oak tree and the puppy, where there are cells deep in the bodies of those organisms that are pretty far away from the environment? And it would be impossible to exchange materials at an adequate rate. So for these large, complex, multicellular organisms, natural selection had to come up with a different solution. How can it be that every cell in an oak tree's body or in a dog's body has access to exchange with the environment? And that solution is a circulatory system. And in a circulatory system, there's some type of fluid, and that fluid moves between a site of exchange with the environment and every cell's immediate surroundings. So In this diagram, the fluid is represented by the purple and the orange arrows, and that fluid circulates between some part of the body where materials are exchanged with the environment, and we'll talk more about that in just a moment, and the fluid that's surrounding every single body cell. So only certain parts of our bodies are intended to exchange materials with the environment. What are those parts? Think about the ways in which your body obtains things from the environment. And by things, I mean gases and nutrients, right? We take in gases and we take in nutrients. And then how does your body give off things to the environment? What are we getting rid of? Waste products, right? All right, so these are our sites of exchange. We have a respiratory surface. In mammals, we're mammals, there are lungs, and at our lungs, we exchange oxygen and carbon dioxide with the environment. This is the respiratory surface in a fish, and remember the fish lives in a watery environment, whereas we live in a gaseous environment. So as blood flows through those capillaries that occur in the fish's gills, that blood gains oxygen right out of the water. The oxygen moves into the blood, and that blood loses carbon dioxide. Carbon dioxide leaves the fish's blood and moves into the water, and that's exchange. Here are our lungs. Our lungs have microscopic air sacs called alveoli, and those air sacs are coated with pulmonary capillaries. So as blood flows through those pulmonary capillaries, that blood gains oxygen and loses carbon dioxide. And of course, when we exhale air that has a higher concentration of carbon dioxide than the air we originally inhaled. So this is our body's way of exchanging gases with the environment, your lungs. Another exchange surface in our body is our gastrointestinal tract, or our GI tract. If you think about what might be inside your GI tract right now, it's a tube that runs the length from your mouth to your anus. So all of the food and liquid that we ingest passes through this tube, and technically the contents of that tube are not considered part of your body. So whatever you ate for breakfast this morning, it's inside the tube, but technically it's not part of your body. But while it's in the tube, we obtain nutrients, water and nutrients from that ingested food. So we will cover the digestive system later this semester, but during a process known as absorption, shown here, number 3, during absorption is when those nutrients have been sufficiently digested, so they're small enough, those nutrients now leave the tube, leave the GI tract, and they enter your blood one way or another. They get into your blood. And now once they're in your blood or your lymphatic system, now those nutrients are considered part of your body. So here we're looking at the lining of the small intestine, and the protein is being chemically digested into smaller and smaller molecules, a peptide, and then finally the building blocks of proteins, which are amino acids. And it's those small molecules, the amino acids, that are able to cross the epithelial lining of the GI tract and move into your blood in this case. And so the process that's being shown here is the absorption of amino acids. And then our third site of exchange is our urinary system. Your kidneys are the urine-producing organs of the body, and they are constantly filtering blood and incorporating waste products into urine. We will talk a lot more about the urinary system this semester, but that urine includes water and waste products like excess sodium ions, urea, bilirubin, creatinine, and other substances, and those waste products are released into the environment every time you urinate. And here we have a schematic diagram of the cardiovascular system. In the center there, you'll see the heart, and the heart is a muscular pump, and it generates pressure gradients that drive the movement of the liquid, called blood, through a set of tubes, and we call the heart, blood vessels, and the blood the cardiovascular system. And the cardiovascular system connects the organs of exchange with all body cells. So the organs of exchange we were just talking about, here are the lungs. This is where gas exchange occurs. Here's the GI tract. This is where water and nutrients are taken in. And then these are the kidneys. This is where waste products get filtered out of our blood and released back into the environment. I like this diagram a lot. It's another schematic diagram. It's not intended to represent any particular organism, but instead it represents all organisms that have a cardiovascular system. And we see the same components here. We see a muscular heart that pumps blood through a series of tubes called blood vessels. And again, the organs of exchange, the respiratory system, the digestive system, and the urinary system are connected via the blood to all body cells. So blood is a transportation medium, just like BART is a transportation medium, where people can get onto the BART at one station and get off of the BART at another station. So because of our cardiovascular system, every cell in our body is able to exchange materials with the environment. Look at this specimen. It is the head and neck of a cadaver, and the blood vessels were injected with a latex type plasticky material, and the other tissues were digested away. And I show you this to emphasize how massive your cardiovascular system is. No cell in your body is particularly far away from a blood vessel. No cell in your body, more importantly, is particularly far away from a capillary. Those are the tiniest blood vessels. And you are a walking bush of capillaries. If we could digest all your other tissues away and just leave your vasculature in place, your loved ones would recognize you. Your facial features would stand out. All right, so your cardiovascular system has three components: the heart, which we'll focus on in this part 1, the blood vessels and the blood, that's part 2 and 3. And so in order to talk about circulation and exchange, we have to revisit body fluid compartments and talk about the specific liquids that are involved in exchange. So in the cardiovascular system, the two most important fluids are blood and interstitial fluid. Blood is a connective tissue, and blood is composed of a variety of cells that are floating or are suspended in a liquid called plasma. And normally this blood occurs only inside the cardiovascular system. So blood normally occurs inside blood vessels or inside the heart. So here's an artistic representation of blood. You see a variety of cell types, which we'll talk about later: red blood cells, white blood cells, platelets, and they're suspended in an extracellular fluid called plasma. It's not really represented here, but you can imagine those cells surrounded by a fluid called plasma. And of course, blood, this tissue, is inside a blood vessel. Our second important fluid in the cardiovascular system is interstitial fluid. Sometimes this is called tissue fluid. Interstitial fluid in this diagram is shown in sort of a pinky lavender color. It's the fluid that surrounds the cells in this tissue. And here the body tissue cells are sort of beigey yellow and they have purple nuclei. And so we're looking at a capillary bed. Again, capillaries are the smallest blood vessels in the body, and we can tell that these are small blood vessels because we can look at the size of the red blood cells inside the capillary, and we can see that the dimensions of the capillary are just a little bit larger than a red blood cell. So the red blood cells have to pass through these tiny vessels in single file. So we know this is a capillary. And the walls of capillaries are leaky. And so during capillary exchange, there is an exchange of material between blood plasma and interstitial fluid. We've seen this diagram before, very early this semester when we talked about body fluids and body fluid compartments, where in the top panel, we see on the right, we see blood inside a blood vessel. In the middle panel, in sort of periwinkle blue, we see interstitial fluid. And then on the right, we see body tissue cells. And then down below, we see bidirectional arrows across each of the barriers. You'll recall that the barrier between blood and interstitial fluid shown here, that barrier is the blood vessel wall, the capillary wall, and there's a bidirectional movement of water and solutes and gases across that wall. And then the barrier between interstitial fluid and cytosol, or intracellular fluid, is the plasma membrane. And we've studied how materials cross that plasma membrane quite extensively. Right, so again, normally all your blood is inside your cardiovascular system, but blood vessels break, and we've all bled, and that bleeding in this cut finger here, there are blood vessels in the dermis of your skin, and the epidermis and the dermis have been damaged somehow, and that blood is leaking out of blood vessels onto the surface of the skin. And on the right, that bruise, a bruise is bleeding that occurs under the skin. So it looks like the skin is intact here, and blood vessels deep to the skin have been damaged, and blood is leaking out, but it can't penetrate through the skin because the skin is intact. And that's what a bruise is. All right, so let's take a closer look at the heart. There is a lot of material to cover on the heart, and I want to present you with an overview of the big picture for where we're going for the duration of part 1. We will talk about how the heart acts as a muscular pump. With every beat of your heart, it generates pressure gradients that drive blood through this continuous loop of blood vessels. And then your heart exhibits what's known as autorhythmicity. That is, it's self-excitable. Your heart can beat, it can generate pressure gradients without any input from your nervous system. So here we're back to that schematic diagram of your cardiovascular system. And we have the heart in the center there, and it's constantly contracting and relaxing and contracting and relaxing. And that alternating pattern, contract and relax, generates pressure gradients that constantly drive the movement of fluid through this closed set of tubes. We'll talk a little bit about how your heart is dynamic, meaning how your heart rate and the contractility of your heart change in response to changing metabolic demands of your body tissues. What happens when you go from a resting condition to you start to exercise. And then speaking of exercise, we know that regular physical activity brings about pretty sweeping changes to your cardiovascular system. And for reasons that are not entirely understood yet, regular physical activity is well documented to reduce the risk of many diseases. All right, so here's a diagram of the heart that is intended to instruct on the pattern of blood flow through the heart. So most students come to physiology class having studied blood flow through the heart. So I'll just give you some reminders that that your heart has 4 chambers. The top 2 chambers are atria and the bottom 2 chambers are ventricles. The atria, the top 2 chambers, receive blood. So blood enters the heart via the atria, and the bottom 2 chambers, the ventricles, eject blood. And the heart keeps oxygen-poor blood separated from oxygen-rich blood. And a very common color convention in diagrams like this is to represent low oxygen blood in blue, and the right side of the heart receives and ejects low oxygen blood. And oxygen-rich blood is represented in red. And the left side of the heart receives and ejects oxygen-rich blood. Here's another diagram for learning purposes. I'm going to ask you to review or learn blood flow through the cardiovascular system as shown here in these images. And then I've also posted 2 short, sweet, very effective videos to help you learn, relearn, be reminded of how blood flows through the heart. And please go and learn that because you will need that information to move forward. All right, so your heart is composed, it's an organ, so it's composed of multiple tissue types, including 2 different types of cardiac myocytes or heart muscle cells. These 2 types of heart muscle cells are called contractile fibers and autorhythmic fibers. The contractile fibers make up most of your heart muscle cells. So if you've ever seen a heart, a real heart from some type of organism, maybe you've dissected a heart, most of those muscle cells are contractile fibers. They're beefy. They have many myofibrils, so thick and thin filaments, remember, make up myofibrils. And they generate force in response to an action potential, just like skeletal muscle cells do. This is what heart muscle tissue looks like when viewed with a light microscope. Heart muscle cells, unlike skeletal muscle cells, heart muscle cells are branched. We can see in this diagram over here that the cells are not just long cylinders, but they're long cylinder-ish, and they branch or they fork. This is a fork in one cell. These are contractile fibers. They make up the majority of cells in the heart. So if you've ever dissected a heart, most of these cells here are contractile fibers. The second type of heart muscle cells are the autorhythmic fibers, and they are cardiac muscle cells. They're just modified. They are pretty wimpy. They have very few myofibrils, and it's not their role to generate a lot of force. That's the role of the contractile fibers. Instead, the autorhythmic fibers are autorhythmic. They're self-excitable. So it's their job to constantly generate action potentials that are then transmitted to the rest of the heart muscle cells, to the rest of the autorhythmic fibers, and to all of the contractile fibers. And as you recall, in muscle tissue, an action potential precedes contraction. So these autorhythmic fibers are responsible for constantly depolarizing, repolarizing, depolarizing, repolarizing. They generate the rhythm that's responsible for the heart's ability to contract on its own. So these autorhythmic fibers, there are far fewer of them than there are the contractile cells. The autorhythmic fibers make up the structures of the intrinsic conduction system of the heart, and those structures are shown here. There are 5 structures in the intrinsic conduction system of the heart: the SA node, the AV node, the AV bundle, the right and left bundle branches, and the Purkinje fibers. And these are all made up of autorhythmic cells, and they are responsible for, in a very rhythmic fashion, generating action potentials and transmitting those action potentials to the surrounding contractile fibers. These autorhythmic fibers are described as self-excitable. They don't have a stable resting membrane potential. Every other type of cell in your body, has a stable resting membrane potential. But these autorhythmic cells do not. They are constantly depolarizing, repolarizing, depolarizing, repolarizing. And I put a picture of a metronome there. A metronome, now I'm sure people use their phones. I'm sure there's an app for that. But back in the day, a metronome was commonly used by musicians to keep a particular pace. And so these autorhythmic fibers, because they constantly depolarize and repolarize on their own, they are what allows the heart to contract without any signals from the nervous system. And they are also responsible for your heart as an organ contracting in a super coordinated fashion. So these autorhythmic fibers, their electrophysiology is actually really cool. We're not going to study it in detail like we studied the electrophysiology of neurons. Autorhythmic fibers have their own electrophysiology, we're not going to study it, but in some later class you might, and you'll be well equipped for that. The way these cells are able to self-excite is because they have no stable resting membrane potential. They have a set of ion channels, voltage-gated ion channels in their membranes that allow their membrane potential to automatically fluctuate like this. So you're familiar with this graph, believe it or not. We've seen this type of graph many times, where on the vertical axis we have membrane potential expressed in millivolts, and on the horizontal axis we have time expressed in seconds. And so we're looking at the change in membrane potential of an autorhythmic fiber over time. Remember, there is no stable resting membrane potential. That's why there's no flat line here ever. Instead, in autorhythmic fibers, we observe what's known as a pacemaker potential. The pacemaker potential is the change in membrane potential that occurs just preceding an action potential. So these cells bring themselves to threshold. And once we achieve threshold, then a different set of voltage-gated ion channels open up, and you can recognize the components of an action potential. There's the depolarization phase, followed by the repolarization phase. And then there's another pacemaker potential. So the intrinsic conduction system of the heart is made up of autorhythmic fibers, those cardiac myocytes that have very few myofibrils and no stable resting membrane potential. They're constantly depolarizing and repolarizing. Shown here are those autorhythmic fibers that have no stable resting membrane potential. They're constantly depolarizing and repolarizing, and that wave of depolarization will flow from one component of the conduction system to the next in a very orderly fashion. So 1, 2, 3, 4, 5. And as that wave of depolarization passes through the intrinsic conduction system, that wave of depolarization is transmitted or passed on to the contractile fibers. And in this way, a wave of depolarization passes across all the heart muscle cells so that the heart can beat as a coordinated unit. If we could remove the rest of the tissues from the heart and only leave the autorhythmic fibers, we'd have a structure that looks like this. Obviously, this is an artistic representation, but this is the intrinsic conduction system of the heart. The sinus node, or the SA node, normally sets the pace and transmits the signal to the atria as well as to the AV node. The AV node transmits the signal to the AV bundle, then to the right and left bundle branches, and finally to the Purkinje fibers. So the SA node is commonly referred to as the pacemaker of the heart. Of all those structures in the intrinsic conduction system, it's the SA node that has the highest intrinsic rate of firing, meaning it has the highest intrinsic rate of depolarization, repolarization, depolarization, repolarization, the fastest rhythm. And typically that's about 100 beats per minute. So as that SA node depolarizes, that wave of depolarization is subsequently transmitted across both atria from one contractile fiber to the next, as represented by the purple arrows here. An action potential is transmitted from one heart muscle cell to the next heart muscle cell at electrical synapses. In this diagram, we can revisit the structure of cardiac muscle cells. And this applies for both the autorhythmic fibers and the contractile fibers. Heart muscle cells are joined together at a structure called an intercalated disc. And so this structure right here is an intercalated disc. And if we open up that intercalated disc, as shown here, we can take a look at the cell junctions holding the plasma membrane of these two neighboring cells together. There are two types of cell junctions that we find in an intercalated disc. The first one is represented up here and these are desmosomes. Desmosomes are often described as spot welds or little snaps that join neighboring plasma membranes together. So these desmosomes ensure that these heart muscle cells stay connected, that they don't tear apart from each other. The second type of cell junction that we find in an intercalated disc is shown down here. They're the gap junctions. And we've talked about gap junctions before. We've talked about electrical synapses before. In one of these heart muscle cells, depolarization is going to be caused by this influx, this inward movement of positively charged particles, those ions. And those ions are able to move from that cell into the neighboring cell through these gap junctions here, these little fluid-filled tunnels that connect the cytosol of neighboring cells. And as those positively charged particles move from one cell into the neighboring cell, it's enough to bring the neighboring cell to threshold. Going back to the conduction system, at the AV node, our second component of the intrinsic conduction system, the action potential that was initiated by the SA node is transmitted from the atrium to the ventricle via the AV node. The AV node is also made of autorhythmic fibers, and if we took those cells and cultured them in a petri dish, we'll observe that they have a slower intrinsic firing rate. They are self-excitable because they're autorhythmic fibers, but they will depolarize and repolarize at a rate of about 40 to 60 beats per minute. And because the SA node has a faster rate of firing, the SA node is the pacemaker because it overrides the intrinsic firing rate, the intrinsic rhythm of all the other autorhythmic fibers. So this is why the SA node is the pacemaker. So we're talking about what's happening here. The SA node fires an action potential, and that action potential, as shown by the purple arrows, is transmitted from muscle cell to muscle cell across both atria. Following that wave of depolarization in the atria contract, and that wave of depolarization needs to be transmitted to the ventricles next. The way that action potential is transmitted between the atria and the ventricles is through this AV node. The AV node is the only passageway for an electrical signal to move between the atria and the ventricles. Not really well represented on this picture, but there is a layer of insulation, electrical insulation, that occurs between the atria and the ventricle, and it's called the fibrous skeleton of the heart. And we'll look at that fibrous skeleton of the heart in just a moment. In this diagram, the way the artist has drawn the heart, we see that this heart is in atrial systole. That means that the atria are contracting, and we can see how the atria are pinched in. During atrial systole, extra blood is moved from the atria into the ventricles. And it's important that the atria contract first in order to push this extra blood into the ventricles. And then later, the ventricles will contract to eject most of that blood out of the heart. So in order to create this delay that allows the atria to contract before the ventricles, the only way in which the action potential can be transmitted from the atria into the ventricles is through the AV node. And the reason for this is the heart contains a layer of insulation. It's made up of fibrous connective tissue, and fibrous connective tissue is not electrically excitable. The wave of depolarization that occurs in the atria does not affect the cells that make up the fibrous skeleton of the heart. And so the fibrous skeleton of the heart is a barrier, an electrical barrier. It's an insulator between the atria and the ventricle. So here we've taken a heart and we've made a transverse section to separate the two atria from the two ventricles. And now we're taking a top-down view. We're looking at all four valves. And also in that picture are the two ventricles. So we see lots of anatomical structures with the word fibrous in them. And all of these make up what's known as the fibrous skeleton of the heart. And they act as electrical insulators, preventing depolarization in the atria from causing depolarization in the ventricles. Instead, it's depolarization of specifically and only the AV node that leads to depolarization in the ventricles. So this is the pathway of the transmission of an action potential between the atria and the ventricles. So it begins with depolarization of the AV node. The AV node transmits the action potential to the AV bundle, to the right and left bundle branches, and to the Purkinje fiber. So there's this wave of depolarization that's spread through the conduction system and onto the contractile fibers of the heart. And so in this way, the ventricles contract after the atria have contracted, and contraction of the ventricles is known as ventricular systole. During ventricular systole, blood will be ejected from both the right ventricle and the left ventricle. So there's this pacemaker chain of command in your heart, and it's based on the intrinsic rate of firing of each of these components of the intrinsic conduction system. So the SA node, as I mentioned earlier, the SA node has the fastest rate of firing, about 100 beats per minute, and this is why it's the pacemaker. All the other components follow its lead, so to speak. And so if the SA node becomes damaged, the AV node will take over as the pacemaker. And so some ways in which the SA node could become damaged, let's say a person could have a heart attack, a myocardial infarction. And during a heart attack, a portion of heart muscle tissue is deprived of oxygen and nutrients, and often those heart muscle cells die and they cease to function. So if somebody suffers a heart attack that affects tissue in their right atrium, it could lead to the death of the cells that make up the SA node. So now this person just lost their natural pacemaker. In that case, the AV node, it's still functioning. It's still spontaneously depolarizing and repolarizing. The AV node will take over now as the rhythm leader of the heart. Should the AV node become damaged, then the chain of command follows. The AV bundle will become the new pacemaker of the heart. And as we move further down the chain of command, we see a slower intrinsic pace, so that those Purkinje fibers have their own inherent rhythm of about 20 to 25 beats per minute. And presumably that's enough to keep somebody alive, maybe a little while hopefully that cardiac muscle tissue can heal, although it has a limited capacity to do that. So when somebody has an irregular heartbeat or an arrhythmia, a very common and pretty simple, successful treatment therapy for that is to install an artificial pacemaker. Pacemakers in general are really simple devices. It consists of a battery and 2 electric leads, and these electric leads will deliver a little burst of electricity in a very rhythmic fashion to structures of the intrinsic conduction system in both the atria and the ventricles. And here we see the batteries implanted here, just deep to the skin, so it's pretty accessible. If the battery needs to be changed, it's a minor surgical procedure. And from the battery, those electrical leads are thread through large vessels and into the heart muscle itself. So pacemakers have gotten more sophisticated recently, in the last decade or so. We now have dynamic pacemakers, also known as activity adjusted pacemakers. And so they have additional devices in them that can determine when you engage in physical activity. So they might have an accelerometer that can detect changes in the movement of your body. And some of them have chemical sensors that detect changes in your blood gases, which would occur with the onset of physical activity. Next, we get to talk about how the nervous system influences both heart rate, how many beats per minute, and how hard your heart contracts with each beat. That's known as contractility. So far we've been talking about how the heart can beat on its own without any input from the nervous system, and the SA node serves as the pacemaker of the heart. As you recall from our coverage in the nervous system, your heart rate and contractility are influenced by the autonomic nervous system. And I hope this diagram looks familiar to you because we used it as an example of an autonomic reflex in our coverage of the nervous system. So let me remind you, in this reflex, there are a variety of sensory receptors and sensory inputs into the cardiovascular center. The cardiovascular center is here. That's the integration center in the medulla oblongata. And the cardiovascular center gives rise to two different forms of motor output: sympathetic motor output and parasympathetic motor output. So these sensory inputs include higher brain centers, so your thoughts, your emotions get fed into the cardiovascular system, as well as sensory receptors that detect body movement, your proprioceptors, chemoreceptors, chemically sensitive cells that occur in some of your large blood vessels, like the aortic arch and the carotid bodies, baroreceptors, these stretch sensitive cells that are monitoring your blood pressure, and they're also located in the aortic arch and the carotid bodies. And so all of these sensory inputs are transmitted to the cardiovascular center, and the cardiovascular center combines them, analyzes them, and determines whether a motor output is required. And here, the motor output, sympathetic or parasympathetic, will influence how fast and or how hard the heart beats. So I hope you recall that when we have an increase in sympathetic stimulation to the heart, the neurotransmitter that gets secreted onto heart muscle cells is norepinephrine, and that's going to have two outcomes. The heart will both beat faster and it will beat harder. And when the cardiovascular center increases parasympathetic stimulation to the heart, the neurotransmitter secreted is acetylcholine. Acetylcholine is secreted onto specific regions of the heart, and it serves to slow heart rate. It has no direct effect on contractility, but it definitely slows heart rate. So how does that happen? We'll start with parasympathetic motor output. How does an increase in stimulation along the vagus nerve cause heart rate to slow? So here's a new diagram for us. In this diagram, the vagus nerve is represented in dark blue. The vagus nerve is a cranial nerve. It emerges from the brainstem, and the vagus nerve innervates many structures in our body, including the heart. Here we see the vagus nerve innervating the SA node and the AV node, and I hope you remember that the vagus nerve secretes acetylcholine, and acetylcholine is going to have the effect of slowing heart rate. So acetylcholine, when it's secreted into the synapse between the motor neuron and the autorhythmic fibers of the SA node or the AV node, acetylcholine is going to bind to and open ligand-gated potassium channels. So let's think more about that. We've seen this picture many times, and we've done a similar exercise. That image represents a cell at resting membrane potential and the concentration gradients across that membrane for sodium, chloride, potassium, and other solutes. So in your mind's eye, insert a ligand-gated potassium ion channel there and open it. It's acetylcholine that causes it to open. So now we have an open potassium channel in that membrane. In which direction is potassium going to move by diffusion? Is more potassium going to leave the cell or enter the cell? more potassium will leave the cell. If more potassium leaves the cell, how does that affect membrane potential? Does the membrane potential become more negative on the inside or less negative on the inside? Potassium leaves the cell. Is the inside more negative or less negative? The inside of the cell becomes more negative, more negative. In this way, acetylcholine is going to affect the steepness of the pacemaker potential. And by steepness, I mean the slope. So here's our pacemaker potential. It has a particular slope, like mathematical slope, rise over run. How steep is that pacemaker potential? Under the influence of acetylcholine, more potassium is going to leave the cell, making the inside of the cell increasingly negative. So moving in this direction, away from zero. That's going to alter the slope of the pacemaker potential. The slope is going to become shallower. So the pacemaker potential is going to extend over time. It's going to have a longer duration. And so in this way, acetylcholine slows heart rate by extending the duration of the pacemaker potential. These heart muscle cells, these autorhythmic fibers, become slower to reach threshold. Parasympathetic motor output is responsible for your resting heart rate. Recall that an average adult resting heart rate is about 70 beats per minute. And also remember that the SA node, the pacemaker of the heart, has an intrinsic rate of firing of about 100 beats per minute. So what slows down the SA node, giving you a resting heart rate of around 70 beats per minute? It's this constant influence delivered by the vagus nerve in the form of acetylcholine. So the SA node is always going to fire, but acetylcholine will slow it down. So your normal resting heart rate is produced by the inherent or the intrinsic rate of the SA node, 100 beats per minute roughly, slowed down under the influence of acetylcholine delivered by the vagus nerve. So when you're at rest and it's appropriate to have a slow heart rate, influence of the vagus nerve onto the intrinsic conduction system of the heart, most importantly the SA node, but also the AV node, helps to extend that pacemaker potential and slow heart rate. And there are times where we need your heart rate to increase. We need to reduce parasympathetic stimulation and increase sympathetic stimulation. And this might occur with emotional excitement or physical activity. So an increase in sympathetic activity to the heart causes heart rate to increase and also causes the heart to beat harder. With every systole, every contraction, more tension is generated. So norepinephrine is the neurotransmitter secreted by cardiac accelerator nerves onto the heart. And norepinephrine is going to bring about changes in both heart rate as well as contractility. So here we can see the cardiac accelerator nerves. These are all postganglionic motor neurons that will synapse on the heart. They synapse many places on the heart, as we'll see. And they all secrete norepinephrine. And again, norepinephrine is going to both speed up heart rate and increase the tension that the heart muscle cells generate with every contraction. In this picture, we can compare parasympathetic innervation of the heart and sympathetic innervation of the heart. Parasympathetic innervation comes by way of the vagus nerve. And in this diagram, we clearly see branches of the vagus nerve innervating the SA node and the AV node. And you remember at these synapses, acetylcholine is going to be secreted and it slows down heart rate. In contrast, we have many, many branches of the cardiac accelerator nerves delivering sympathetic stimulation. And at all of these synapses, norepinephrine is secreted. When norepinephrine is secreted here at the SA node and at the AV node, that's going to have the effect of increasing heart rate. We'll take a look at that in a moment. When norepinephrine is secreted here over the rest of the myocardium, it's going to have the effect of increasing contractility. In response to norepinephrine, these heart muscle cells, these contractile fibers, will generate more tension with every contraction. So norepinephrine has two different effects on the heart. It's going to increase the firing rate of the SA node, and it will increase the contractility of those contractile fibers, so the heart will beat more forcefully. To understand how norepinephrine affects heart rate and contractility, we need to revisit cell-to-cell communication. Shown here is a sympathetic motor neuron and a cardiac myocyte. We'll start by talking about this cardiac myocyte as if it's an autorhythmic fiber, because norepinephrine is going to influence heart rate by influencing the autorhythmic fibers. And then later on, we'll revisit this image and we'll think about the cardiac myocyte as a contractile fiber, because norepinephrine is also going to influence the force with which this muscle cell contracts. So, thinking about this heart muscle cell as an autorhythmic fiber. The autorhythmic fiber has receptors for norepinephrine. Norepinephrine binds to these receptors and initiates what I hope you recognize as a signal transduction pathway inside the cell. Importantly, this signal transduction pathway makes the membrane more permeable to calcium, and calcium moves into the cell. Here we are with the same image. I want to use this image to have you understand how calcium influences membrane potential in an autorhythmic fiber. So in your mind's eye, please insert into the membrane a calcium ion channel and open it. It's not shown here, so I have to tell you, there is more calcium outside the cell than inside the cell. So in which direction is more calcium going to move through that open ion channel? Is more calcium going to leave the cell or enter the cell? Calcium is going to enter the cell down its electrochemical gradient. How is the influx of calcium going to influence the membrane potential of this cell? Is the inside of the cell going to become more negative or less negative? Remember, calcium's valence is +2. It's a double positive charge particle. So with this influx of calcium into the cell, the inside of the cell is going to become less negative. Yeah? And we're back to this image. Under the influence of norepinephrine, the slope of the pacemaker potential is going to change. Remember, the slope is the steepness of this line. Under the influence of norepinephrine, as calcium moves into the cell, the pacemaker potential slope increases. It becomes steeper. And this means the autorhythmic fiber is quicker to reach threshold. The pacemaker potential is shorter in duration. And in this way, norepinephrine increases heart rate by shortening the duration of that pacemaker potential. So norepinephrine increases the intrinsic rate of firing of these autorhythmic fibers, and it does this by opening calcium channels, allowing calcium to move into the cell, and now that membrane potential reaches threshold more quickly. But there's a second effect of norepinephrine on the heart, and that affects mostly the contractile fibers, so those bigger, beefier cells that do most of the physical work of the heart. It causes these muscle cells to contract with greater force. So here we are back at this picture of cell-to-cell communication, where a sympathetic motor neuron meets a cardiac myocyte. Only now we need to think of the cardiac myocyte as a contractile fiber. And once again, we see that this cardiac myocyte has receptors for norepinephrine. And when norepinephrine binds to its receptors, that triggers a signal transduction pathway. We've previously talked about that signal transduction pathway having this effect. It causes the plasma membrane to become more permeable to calcium. So calcium enters the cytosol. This is a muscle cell. Calcium in the cytosol is very important for muscle contraction. Also shown here is the release of calcium from the sarcoplasmic reticulum. Under the influence of norepinephrine, there is both an influx of calcium into the cell from the cytosol, as well as a release of calcium into the cytoplasm from the sarcoplasmic reticulum. So under the influence of norepinephrine, the amount of calcium that enters the cytosol is much greater than when the cell is not under the influence of norepinephrine. This leads to a greater force of contraction, also known as higher contractility. We've seen this image before in a different chapter. We looked at this image when we were understanding the role of calcium in muscle contraction in skeletal muscle. It's not too different in cardiac muscle. So you remember in skeletal muscle that an action potential causes calcium release channels in the sarcoplasmic reticulum to open up. And as calcium floods the cytosol, it binds to troponin, which is a protein that's part of the thin filament. When calcium's bound to the thin filament, it exposes the myosin binding site on actin, and the contraction cycle can begin. So the more calcium we have in the cytosol, the more myofibrils will engage in the contraction cycle, and the greater the force of contraction will be. So we can sum up the effects of norepinephrine and also remind ourselves that norepinephrine is also secreted into the blood from the adrenal medulla. So up top, this is what we've just been talking about, the effects of norepinephrine on the heart, right? Norepinephrine, when it's secreted by this postganglionic sympathetic motor neuron, norepinephrine causes the heart to beat faster and to beat harder. Well, norepinephrine is also secreted into the blood by the adrenal medulla in response to sympathetic stimulation. So norepinephrine is norepinephrine. It's the same signaling molecule, and it will bind to receptors whenever it encounters those receptors. So the source of the norepinephrine, whether it was secreted at a synapse or it was secreted into the blood, that norepinephrine is going to lead to the same behavioral changes in the heart. The heart rate will speed up and the heart contractility will increase. Revisiting this picture, we can focus on what's labeled here as sympathetic cardiac nerve. These are branches of the cardiac accelerator nerves. And the artist was careful to show that it innervates the SA node, the AV node, as well as many different points on the myocardium. So the secretion of norepinephrine at the SA and the AV node will help increase heart rate, and secretion of norepinephrine widely across the myocardium will help increase heart contractility. Going back to this diagram just one more time, we've worked with this diagram quite a bit across two different organ systems, and I hope now you've been able to fill in some mechanistic details. You have a better understanding of how acetylcholine can slow heart rate and how norepinephrine can speed up heart rate as well as contractility. Our next topic is the ECG, the electrocardiogram. An electrocardiogram is a record of the electrical events that occur in the heart. And electrocardiogram is often abbreviated as ECG or EKG. You could use either term. EKG is an older term, it's the original term. where the K comes from the German. So I like this diagram. It's fairly elegant and it allows one to very easily match up the events in an ECG recording to the electrical events in the heart. So let's look at the ECG recording up top. An ECG recording includes 5 components: a P wave, the QRS complex, and a T wave. All of those marks occur during one cardiac cycle, so during one heartbeat. And the artist used shading to illustrate down below the electrical events that are occurring in the heart that produce this particular characteristic ECG trace. So during the P wave here, the SA node fires an action potential, and that electrical stimulus is transmitted throughout both atria. As the atria depolarize, it's recorded as a P wave on the ECG. Next, we have the electrical impulse being transmitted from the atria into the ventricles, and then the ventricles are going to depolarize. And depolarization of the ventricles gives us this much larger event, the QRS complex. On the ECG recording, the magnitude of deviation from the isoelectric line represents the overall quantity of muscle cells that are depolarizing. There are far more muscle cells in the ventricles than there are in the atria. So when the ventricles depolarize, it looks like a larger event on the ECG. So QRS corresponds to depolarization of the ventricles. And then we have this T wave. During the T wave, the ventricles are repolarizing. So they've already depolarized and now they're repolarizing, and that change in the electrical behavior of this organ is recorded as a T wave on an ECG. This diagram also allows us to line up the electrical events that occur in the heart with the corresponding recording we observe on an ECG. So we'll start over here. At the onset of depolarization in the SA node, that wave of depolarization then spreads throughout both atria. And during that spread of depolarization in the atria, we observe a P wave on the ECG. Then that wave of depolarization is spread from the atria to the ventricles, and eventually both ventricles depolarize. And ventricular depolarization is observed as a QRS complex on an ECG recording. And then the ventricles are going to repolarize, and that repolarization of the ventricles is observed as a T wave on an ECG recording. So the components of an ECG recording include the P wave, atrial depolarization, the QRS complex, ventricular depolarization, and the T wave, ventricular repolarization. I've included additional explanatory diagrams to help you learn about ECGs. It's good to have multiple representations of the same thing. All right, so the deflections from the isoelectric line that we observe in an ECG, those deflections, just like in an EMG, those deflections are the result of electrical activity in millions of cells. So we are not observing an action potential in just a single cell, but the combined electrical activity of the heart muscle as an electrical organ. On the next few slides, I'll introduce some vocabulary, some conditions that we can diagnose or identify using an ECG recording. The first one is a normal sinus rhythm. That's a good thing. That's what we hope to see when we look at an ECG recording. And normal sinus rhythm refers both to resting heart rate as well as the regularity of the rhythm of the heart. And so a normal resting heart rate is between 50 and 100 beats per minute, and the heart rate is regular. On this diagram of an ECG, we only see one cardiac cycle, so we only see one R peak, but I'll show you on another image. In a normal sinus rhythm, the distance between R peaks. The R to R interval is regularly, meaning depolarization of the ventricles occurs at an even, equal pace. So there is a regular rhythm to the heart rate. And also, in a normal sinus rhythm, there's a P wave, an identifiable P wave on the ECG. And when we see that P wave, we know that the atria have depolarized in advance of the ventricles. So all of those things are good. We can use an ECG to identify an arrhythmia. This is an irregular heartbeat. And an arrhythmia can easily be identified by examining the RR interval in an ECG trace. So the RR interval is the distance from one R to the next R. And we can count up those squares. We can measure how long that distance is. Notice that it's a little bit longer here, and the RR interval is even longer here. And so this person has an arrhythmia. Tachycardia refers to having a resting heart rate over 100 beats per minute. And we can identify tachycardia by looking at the RR intervals. They're very short, indicating that the heart is beating very quickly. They appear to be regular, so this person does not have an arrhythmia, but they are tachycardic. And then the opposite of tachycardia is bradycardia. Bradycardia means having a resting heart rate below 50 beats per minute. And so here we can identify that bradycardia by this very long RR interval, telling us that the heart rate is very slow. And then there's fibrillation. This is bad, bad, bad. Fibrillation means that the heart muscle is contracting in a very uncoordinated fashion. It might be viewed as like a jiggling mass of muscle tissue. And this means that the heart is not acting as an effective pump. In order for the heart to work as an effective pump, it needs to contract and relax in a very coordinated fashion. So when the chambers of the heart are relaxed, they're allowed to fill with blood. And when the chambers of the heart contract, they move that blood into the next vessel or chamber. And if the heart is contracting as this wiggling mass, then that filling and ejecting is not happening adequately. And this can mean death for the patient pretty quickly. And so fortunately, we have this pretty simple life-saving device known as a defibrillator. If you've been trained in CPR, you learned how to use a defibrillator. You probably learned how to use an automated external defibrillator. Not these paddles as shown here, but how a defibrillator works is it delivers a jolt of electricity across the skin, and that serves to simultaneously depolarize all of the heart muscle cells at once. It's almost like the reset button for this electrical jiggling mass of heart muscle tissue. If all of those cells can depolarize all at once, then suddenly all of those cells are on the same page, so to speak. And it allows the SA node to resume its role as the pacemaker of the heart. And we hope the electrical conduction system falls in line. So AEDs, automated external defibrillators, are these portable devices that you probably learned how to use when you were certified in CPR. They are so important and so life-saving. They're in all sorts of public buildings. They're on campus, they're in stadiums, they're in restaurants, and they're very, very user-friendly. They will diagnose the arrhythmia, the defibrillation, I should say, and only when necessary will the device deliver a charge of electricity or defibrillate the patient. There are also defibrillators that can be surgically implanted into the patient. And so the typical recipient of an implanted defibrillator has a problem with their intrinsic conduction system, and they are likely to go into fibrillation, which is a life-threatening situation. So an implanted defibrillator is able to detect the activity of the electrical conduction system and to know when the heart has gone into fibrillation and immediately counter that by delivering electrical stimulation by acting as a defibrillator. And so again, this electrical stimulus brings all of the heart muscle cells into a state of depolarization at the same time. So it's the reset button, And we hope that the patient's intrinsic conduction system, the SA node specifically, takes over. So it's a life-saving device. These devices currently are able to send data to the physician's office. So every time they're triggered, every time they go into action, the physician gets that data via the internet. Okay. We're going to spend the next few minutes talking about the cardiac cycle. And the cardiac cycle are the events that occur in the heart during just a single heartbeat. So it's really important that you understand the pathway that blood flows through the heart. If you need to review it, you can use this diagram or watch videos on the internet. I'm going to move forward as though you do understand the pathway that blood flows through the heart. I hope you remember the pulmonary circuit and the systemic circuit. So the heart is a muscular pump, and in the pulmonary circuit, the right side of the heart pumps blood to the lungs and back to the heart. And in the systemic circuit, the left side of the heart pumps blood to the entire body and back to the heart. So the cardiac cycle is a sequence of events, highly coordinated events that occur during one heartbeat. And we're going to take a pretty detailed look at the events of the cardiac cycle. They are overlapping, and this might be new vocabulary for you. So diastole means relaxation, and systole means contraction. So in the cardiac cycle, we can begin with atrial diastole. The atria are relaxed, and they're receiving blood, and then they contract. That's atrial systole, and they move some volume of blood into the ventricles. The ventricles are relaxed. They're receiving blood that's flowed through the atria and into the ventricles, and then the ventricles contract. That's ventricular systole. During ventricular systole, blood is ejected into the aorta from the left side of the heart and into the pulmonary trunk on the right side of the heart. The events of the cardiac cycle are a cycle, so anywhere we start, we're going to end up there. And we're going to take a detailed look at the cardiac cycle. This is a great diagram, and I'll encourage you to spend a bunch of time with it. There's a lot of information to be had pretty easily here. So again, we could start anywhere in the cardiac cycle and end up there. I'm going to follow the lead of the artist and move through it 1, 2, 3, 4, 5. But before we do that, I want to draw your attention to the 2 concentric circles that are in the middle of the diagram. And the deeper circle, the smaller circle, represents or shows us when the atria are contracting, that's atrial systole, shown in red, and when the atria are relaxed, that's atrial diastole, shown in peach. And then just superficial to that, the larger circle shows us what's happening in the ventricles. So when the ventricles are relaxed, that's represented by peach, and that's ventricle diastole. And when the ventricles are contracting, that's ventricular systole, that's represented in purple. So let's start at the first position. So we're going to jump into this cycle in late diastole. All 4 chambers are relaxed, and the ventricles are filling passively. So in that diagram, number 1, the artist uses arrows to show the flow of blood in the heart. So all 4 chambers are relaxed. We have blood entering both the right atria and the right ventricle. We have blood entering both of the atria, the right atrium and the left atrium. And that blood is just passively dropping down into the ventricles. The AV valves are open and blood is filling the ventricles. And now we move on to panel number 2. This is atrial systole. So the atria are contracting, and that's going to drive a few more milliliters of blood into the ventricles, and the black arrows show the blood moving into the ventricles. In panel 3, this is a very brief portion of the cardiac cycle, brief but important. It's called isovolumetric ventricular contraction. That's a mouthful. So ISO, what does ISO mean? ISO means same. Isovolumetric. Oh, I see on this diagram it says volumic. That's okay. Another term for it is isovolumetric. Isovolumic or isovolumetric means the volume of the blood in the ventricles doesn't change. So for a brief window of time, all 4 valves are closed. The 2 AV valves are closed, the tricuspid and the bicuspid, and the semilunar valves are closed. So there's no blood entering the ventricles and there's no blood leaving the ventricles. This is why it's isovolumetric ventricular contraction. So the ventricles have just started to contract. They've generated enough pressure, they've increased the pressure in the ventricles to close the AV valves, but not enough pressure to open the semilunar valves. So the ventricles begin to contract, pressure inside the ventricles rises as those walls squeeze the blood, and as that pressure goes up, the AV valves shut. This prevents the blood from moving from the ventricles back up into the atria. That wouldn't make for an effective pump. We want to make sure the blood keeps moving in one direction. And so the AV valves close, preventing the backflow of blood up into the atria. Eventually, the semilunar valves, the aortic valve and the pulmonary valve, are going to open, and blood will be ejected from the ventricles into the aorta and the pulmonary trunk. But that hasn't happened yet in panel 3. We're still isovolumetric because all 4 valves are closed. So let's move on to panel number 4. This is ventricular ejection. So the pressure generated by the contracting ventricles has become high enough to force open the semilunar valves. And so blood that's in the right side of the heart is ejected through the pulmonary semilunar valve into the pulmonary trunk, and blood that's in the left ventricle passes through the aortic semilunar valve and into the aorta. And so now blood is leaving the ventricles. As we'll look at in a few minutes, we'll look at how the volume of blood in the ventricles changes across the cardiac cycle. During ventricular ejection, the volume of blood in each ventricle declines. And moving on to the 5th panel, this is isovolumetric ventricular relaxation. So there's that word again. I say isovolumetric, and that term is in lots of textbooks. Wherever I got this picture, they use isovolumic. It just means that the volume in the ventricles stays the same for this very brief window of time. All 4 valves are closed. So the ventricles have relaxed, the atria are still relaxed. Blood is flowing into both of the atria, but it's not sufficient to reopen those AV valves. As more blood enters the atria, eventually the pressure in the atria is sufficient to overcome and open up those valves. And that brings us back to panel number 1. We're still in diastole. The atria are relaxed, the ventricles are relaxed. This is sometimes referred to as the relaxation period, and passive filling is happening. Blood is flowing freely into the atria and through the open AV valves into the ventricles. And we're back where we started again. The way the cardiovascular system is structured is to cause the one-way flow of blood all the time. Blood flows in just a single direction 24 hours a day. And it's important that blood flows in just one direction. Blood is always moving forward, because any backward movement of blood, any backflow, means that the cardiovascular system isn't being effective. It's not effectively circulating the blood. And so the way we get blood to move is the combined effects of pressure gradients and one-way valves. So pressure gradients. A pressure gradient occurs when there's an area of high pressure and it's connected to an area of low pressure. And as we briefly spoke of in the respiratory system during lab, air will flow from an area of high pressure to an area of low pressure. Liquid, likewise, will always flow from an area of high pressure to an area of low pressure. And you do this every day when you squeeze toothpaste onto your toothbrush. So toothpaste is a liquid. It's a very viscous liquid, but it's a liquid. And when you apply pressure to the tube, you're increasing the pressure inside the tube. As a result of that pressure increase, the toothpaste comes out of the tube. It moves from an area of high pressure to an area of low pressure onto your toothbrush. And so if we look at this very simple diagram of the left ventricle, the aortic semilunar valve, and the aorta, we see that there are two different pressures shown there. Pressure 1 is inside the left ventricle, and pressure 2 is the pressure inside the aorta. And in order to get blood to move out of the ventricle and into the aorta, we need to establish a pressure gradient. So during ventricular systole, as the walls of the ventricle contract, they increase P1, and only when P1 is higher than P2 will blood leave the ventricle and enter the aorta. And during ventricular diastole, when the ventricle eventually relaxes, the polarity of that pressure gradient will reverse. P1 will come to be lower than P2. Under that instance, the aortic semilunar valve here will close, and that will prevent the backward flow of blood from the aorta into the ventricle. So blood is ensured for forward movement by the combined effects of pressure gradients and one-way valves. There are 4 one-way valves in the heart, 2 semilunar valves and 2 AV valves. And when they're open, they allow blood to flow in the forward direction. When they close, they prevent blood from flowing in the reverse direction. And valves are always opened or closed by pressure gradients. During the cardiac cycle, we can measure changes in pressure. We can also measure changes in volume as blood fills the heart and is ejected from the heart. So in the top panel, we see pressure expressed in millimeters of mercury. And let's just take a quick look at the left ventricle. So think about pressure in the left ventricle over the course of the cardiac cycle. So down here, where pressure in the left ventricle is relatively low, that must mean that the walls of the ventricle are relaxed. So this is ventricular diastole. Then, as the ventricle begins to contract, pressure gets very high. This is ventricular systole, when the ventricles are contracting. And then as the ventricles relax once again, that pressure falls and goes back to its low ventricular diastole level. In the bottom panel, we can also look at changes that occur in the amount of blood inside the left ventricle over the course of the cardiac cycle. And so this is volume in blood expressed in milliliters. So during the relaxation period here, the ventricle is relaxed, it's not contracting, and the quantity of blood inside the left ventricle is increasing. This occurs as blood moves through the atria and into the ventricle. And then we see this pretty precipitous drop in volume in the left ventricle. So this is ventricular ejection. So we know that the semilunar valve has opened and blood is being shot out of the ventricle and into the aorta. And then we see this period of filling again. So we know the wall of the ventricle must be relaxed and blood is falling in from the atria into the ventricle. So there's a bunch of vocabulary here related to the cardiac cycle. So this is the same diagram that we were just looking at, and now it's labeled to indicate important time points and changes in volume that occur during the cardiac cycle. So point number 1 in the upper panel, we're examining pressure changes in the left ventricle. At point number 1, this is the onset. Onset means beginning. So at point number 1, this is when ventricular systole begins. And I wish I could make that arrow actually a little more precise. As soon as this line starts to go up, we know the ventricle has started to contract because the pressure is rising. At point 2, this is the beginning of ventricular diastole, or the onset of the relaxation period. The ventricles will relax, and that causes the pressure in the ventricles to decline. Moving down to the bottom panel, we have 3 volumes to learn about. The first one is the end diastolic volume. So that's right here. The end diastolic volume is the amount of blood in milliliters that is in the left ventricle or the right ventricle, either ventricle. They hold the same volume of blood. So the end diastolic volume is the quantity of blood that's in the ventricle at the end of diastole, so the end of ventricular relaxation. Contraction is just about to happen. So what's the maximum amount of blood before the ventricle contracts during this particular cardiac cycle? That's the end diastolic volume. And you can see it's the highest point on this graph, right? At no other point does the ventricle contain more blood than it does at the end diastolic volume. Number 4 shows us the stroke volume. The stroke volume is the volume of blood that is ejected from a ventricle during one cardiac cycle. So during one contraction of the ventricle, how much blood is shot from the ventricle into, in this case, the aorta. And so the difference between 0.3 and 0.5 is the stroke volume. 0.5 is the end systolic volume. How much blood remains in the ventricle when the ventricle finishes contracting and starts to relax again. Your ventricles are never completely emptied. Always some blood remains, and the quantity of blood that stays in the ventricle when that ventricle finishes contracting and starts to relax again is called the end systolic volume. These volumes are malleable, they're mutable, they change according to the metabolic demands. Okay, so here are those definitions written out in words, along with some representative values. So the end diastolic volume is the final quantity of blood that's in the ventricle when they first begin to contract. The stroke volume is the amount of blood that is ejected from the ventricle with every beat. It's about 70 milliliters on average. And then the remaining amount of blood is the end systolic volume, the volume of blood remaining in each ventricle at the end of contraction, the end of ventricular systole. So here's a little math. So if we take the end diastolic volume, that's the maximum amount of blood in the ventricle at the onset of contraction, and subtract the stroke volume, that's the quantity of blood that is ejected during contraction. The amount of blood that remains in the ventricle is the end systolic volume. And then here are those typical values. So end diastolic volume is roughly 130 milliliters. With contraction of that ventricle during that particular cardiac cycle, the stroke volume is 70 milliliters. That leaves the end systolic volume at 60 milliliters. So how much blood does your heart pump? How much blood does your heart pump per minute? That's cardiac output. We'll talk more about that later. How much blood does your heart pump with every beat? That's stroke volume. We just talked about that. We're going to continue to talk about that because stroke volume is dynamic. It's not fixed or static. It can change from one beat to the next beat. So on this heartbeat, perhaps your heart pumps 70 milliliters of blood. On the very next heartbeat, it might pump 80 milliliters of blood. So 70 and 80 are both stroke volumes. So how can it be that the heart can have a different stroke volume from one beat to the next? One thing that influences stroke volume is the length-tension relationship, and we'll look at that in just a moment. But I need to tell you first about the Frank-Starling law of the heart. So these are two different individuals, last name Frank, last name Starling. There's no person named Frank Starling. It's two different people. And they made independent observations that the heart will always eject the same volume of blood that it receives. So during one heartbeat, during one cardiac cycle, if 70 milliliters of blood are added to the ventricle, on the very next contraction, 70 milliliters of blood will be ejected from the ventricle. And if during the next cardiac cycle, 80 milliliters of blood are received by the ventricle, then 80 milliliters of blood will be ejected by the ventricle. The ventricle will contract more forcefully in order to ensure all 80 milliliters are ejected. Again, this is known as the Frank-Starling law of the heart, and we're going to talk about the mechanism. Well, how can it be? How does the ventricle, air quote, know? How does the ventricle know to eject 70 milliliters or 80 milliliters? And so the Frank-Starling law of the heart has to do with the length-tension relationship. And I hope you remember the length-tension relationship from skeletal muscle. We'll review that in a moment. But in essence, here in the heart, the more the ventricles are stretched as they fill with blood, the greater the force of contraction on the next contraction. So the outcome of the Frank-Starling law of the heart is that during systole, the ventricle will eject the same amount of blood that arrived there during diastole. We've seen this image before. This depicts the length-tension relationship in skeletal muscle tissue. It looks different in heart muscle tissue, but I wanted to take a moment and review what the length-tension relationship is, what it means, with something that you're more familiar with before showing you what it looks like in the heart. So let me remind you, on the vertical axis, we have tension developed, expressed as a % of maximum. So how hard can the muscle cell contract? Can it contract at 40% of its maximum, at 80% of its maximum, or can it achieve 100% of its maximum? And on the horizontal axis, that's the length of the sarcomere when contraction begins. So according to the length-tension relationship, when we start off with previously shortened sarcomeres, then the muscle cell is not able to achieve its maximal contraction. Here at this labeled point, the muscle is only able to achieve 80% of its maximum. And then on the opposite side of maximum, if we begin with a sarcomere that's already stretched, it too is not able to achieve its maximum. There's just not enough overlap between the thick and thin filaments to make that sarcomere shorten. And this happens when a muscle is stretched at the beginning of contraction. But in the middle here, when we start with a sarcomere at its optimal length and that cell contracts, that cell is able to generate its peak force. It's able to reach 100% of its maximum. And this is because there's just the right amount of overlap between thick and thin filaments, and there's also enough room for these thin filaments to slide toward the M line. So this is the length-tension relationship in skeletal muscle tissue. The shape of the line here is different in heart muscle tissue, but the factors are still the same. So this is what the length-tension relationship looks like in cardiac muscle cells. Notice the shape of the line, that upward sloping, sort of asymptoting line before it declines. Cardiac muscle cells have a much different length-tension relationship. I want to draw your attention to these axes. I've been telling you that this is the length-tension relationship, but the axes are labeled differently. On the vertical axis, we have stroke volume, but what if we crossed out stroke volume and wrote maximal tension as expressed as a % of maximum, just like on the previous slide? And on the horizontal axis here, what if I cross out ventricular and diastolic volume and we write sarcomere length at the onset of contraction? Then we have the length-tension relationship. And so in these cardiac muscle cells, if we begin with sarcomeres that are already short, we can see that this heart muscle cell is not going to be able to achieve very high tension. Maybe that's 10% of its maximum. And then if we have this normal resting length, this heart muscle cell will be able to generate, maybe this is 50% of its maximum tension. As the sarcomeres stretch, the heart muscle cell will now be able to finally achieve 100% of its maximal tension. And so as the sarcomeres get longer, the force of contraction increases, but only up until a point. When we go beyond some threshold point here, as we continue to elongate the sarcomere, so the sarcomeres are even more stretched, then the amount of tension that this muscle cell is able to generate starts to decline. The heart can't beat as forcefully. So this is the length-tension relationship in cardiac muscle cells. Now let's go back and revisit these axes, but we can use the axes as the artist originally intended. So now our axes are stroke volume on the vertical axis and end diastolic volume on the horizontal axis. So let me remind you what those words mean. Stroke volume is the quantity of blood that's ejected from the heart during one contraction. And end diastolic volume is the volume of blood that's in the ventricle right when it starts to contract. And so that volume of blood in the ventricle is going to determine sarcomere length at the onset of contraction. So down here, let me switch colors. Let's go to blue. Down here, when we have relatively little blood in the ventricle, the sarcomeres are not stretched at all, and the force of contraction is relatively low. In the middle here, as we have a moderate volume of blood in the heart, the sarcomere length is a bit longer, and we have a moderate force of contraction. As our end diastolic volume goes up, so now the ventricle has received more blood during the relaxation period, these sarcomeres become stretched. And in response to this stretch, the heart is able to beat with more force. And so it's this length-tension relationship that allows the Frank-Starling law to happen, that phenomenon. That the greater the end diastolic volume, that's down here, the greater the EDV, the greater the force of contraction, which shows up as stroke volume. The harder the heart contracts, the more blood it ejects. And of course, going back here, there's a limit. So a heart can be overstretched where it becomes a less efficient pump. All right, cardiac output. We're going to continue to talk about the rate at which your heart pumps blood. And that rate is dynamic. It changes from moment to moment. And here we're looking at images from a local race called Beta Breakers. Maybe you're familiar with it. Probably won't happen this year, unfortunately. But think about the person's heart rate at 3 points in the event. Not their heart rate, excuse me. Think about how much blood their heart is pumping per minute. Before the race starts, there's just generally a lot of hanging out and waiting. And then during the race, and some parts of the course are running uphill. And then after the race, this person's relaxed and happy. Think about the rate at which the heart is pumping blood at these 3 points in time, before, during, and after. So we're thinking about cardiac output, and that's defined as the volume of blood that's pumped by a ventricle, either the right or the left, in 1 minute. And cardiac output is very easy to calculate. Cardiac output is determined by multiplying stroke volume times heart rate. So stroke volume, how much blood does your ventricle pump with each beat? And then heart rate, how many beats per minute? All right, so cardiac output is expressed in milliliters per minute. Stroke volume is expressed in milliliters per beat, and heart rate is expressed in beats per minute. These are typical volumes for a body that's at rest. So a body at rest might have a stroke volume of 70 milliliters per beat and a heart rate of 75 beats per minute. If we do that math, the calculated cardiac output is 5.25 liters per minute. And what does that number mean, 5.25 liters per minute? It means that for this person, when their body is at rest, their heart is pumping 5.25 liters of blood every minute. And every body at rest has a cardiac output of roughly 5 liters per minute. So cardiac output is dynamic. Again, it's changeable, and there are several factors that are variables that lead to a different cardiac output. So if we need to increase cardiac output, let's say we just started the race and we need to increase cardiac output, we can increase stroke volume, we can increase heart rate, or we can increase both. And all of those will lead to greater cardiac output. So here are some values during moderate exercise. So stroke volume increases, it almost doubles to 130 milliliters per beat. Heart rate doubles to 150 beats per minute. We multiply those together and we get a cardiac output of 19.5 liters per minute. So this is moderate strenuous activity. So what is your maximum cardiac output? When you're moving physically as much as you possibly can, and your heart is beating as fast and as hard as it possibly can, what's your cardiac output at that point? What's your stroke volume? What's your heart rate? Multiplying those together will give us your maximal cardiac output. We can then use that along with your resting cardiac output to determine what's known as your cardiac reserve. Your cardiac reserve is the difference between your maximum cardiac output and your resting cardiac output. Cardiac reserve is a measure of what your heart is capable of, how dynamic it is. So for an untrained, otherwise healthy individual, cardiac reserve is 4 to 5 times resting cardiac output. So this applies to the example we just went through a moment ago, where this person's resting cardiac output was 5.25 liters per minute. And then we had them exercise. And let's now say that they're exercising at their maximal capacity. Their heart is beating as hard as it can beat, and their stroke volume is also maximized. Whatever their cardiac output is at their maximum is going to be between 4 and 5 times their resting cardiac output. So in the math before, it was got up to 19.5 liters per minute. In a trained endurance athlete, the cardiac reserve can be much higher. Their cardiac reserve can be between 7 and 8 times their resting cardiac output. And we'll talk about some of the effects of endurance training on the cardiovascular system in a little bit. In the opposite direction, we can think about a person who has severe heart disease. For example, someone with late stage congestive heart failure. Someone with late stage congestive heart failure, that means that their heart is not able to pump very much blood at all. It's not an effective or an efficient pump, and they have very little to no cardiac reserve. So if somebody has 0 cardiac reserve, it means their cardiac output while they're at rest is as good as it gets. And so it severely limits their physical activity. And some of the most extreme cases, just going from a sitting position to a standing and walking motion is more than the heart can handle. The heart's not able to pump enough blood to support more physical activity than that. So cardiac output changes from moment to moment throughout the day, 24 hours, 7 days a week. Your autonomic nervous system is modifying your cardiac output. And as part of homeostasis, I like to view homeostasis through this lens. It's a constant shifting balance. A little more, a little less, a little higher, a little lower. So as your cardiac output is being altered throughout the day, you are probably not aware of it. It's being altered by a set of autonomic nervous system reflexes, and so you don't have great conscious awareness over it. So in this diagram, we see the factors that influence cardiac output. And we'll start at the bottom and work our way up. I promise you, you are already familiar with all of these factors. So at our next level here, nope, that's not our next level. At our next level here, this level, these are the two factors that are multiplied together to determine cardiac output. And each of these are variable, right? Stroke volume and heart rate. Stroke volume is how much blood is pumped with each beat, and heart rate is how many beats are there a minute. And both of those are variable. If we go up to the next level, we can see what influences stroke volume and heart rate. And you're familiar with all of these terms. Let's look at stroke volume. So stroke volume is directly related to contractility. So the force with which the heart contracts. When the heart contracts harder, stroke volume goes up. Well, what causes the heart to contract harder? Oh, look, hormones like norepinephrine and influence from the autonomic nervous system via cardiac accelerator nerves. So those are two sources of norepinephrine, and they both can increase contractility. All right, well, what modifies your heart rate? Well, the same thing. You remember that sympathetic motor output causes heart rate to increase, and that norepinephrine can be delivered via sympathetic motor neurons or via the blood, because that norepinephrine was secreted by the adrenal medulla. And so here we are again at this very familiar image where I want to make the point that all day long there's this constant shifting balance between parasympathetic stimulation and sympathetic stimulation. And that's the way that the autonomic nervous system modifies your cardiac output from moment to moment. It's well established. There is a mountain of scientific evidence that regular physical activity promotes longevity, health, and well-being. It lowers the risk of a very long list of disorders and diseases. What that mechanistic relationship is, why does regular activity promote health, is not clearly understood. We're still working that out, and it's multifactorial. It's a complicated picture, and people are right now trying to understand it. What we have worked out, what we do have a better understanding of, are some of the anatomical and physiological effects of regular physical activity on the heart and the cardiovascular system. So we're narrowing our lens on the heart and the cardiovascular system, and what are the effects of regular exercise. So what are the effects of regular exercise on the heart? Let's start by defining what we mean by exercise. There's many different types, and the type that we're focused on here is aerobic activity. And a common definition of aerobic exercise is working major muscle groups for 20 minutes or longer. So aerobic activities include dancing, running, cycling, swimming, anything that works your major muscle groups for 20 minutes or longer. We're going to see an increase in heart rate, and we're also going to see an increase in respiratory rate. So maybe you've had the pleasure of observing changes in your own body as you take up a new form of aerobic activity. Let's say you take up running, and in the beginning, you can barely run for 5 minutes without having to stop, rest, catch your breath. But you keep at it. You're running every day, every few days, and eventually you're able to run for 10 minutes and then half an hour. And over time, you run marathons, and you're able to run for several hours now without taking a break. Why? What changes have occurred in your body? Well, all of the changes support your skeletal muscle cells' ability to produce ATP and augment or improve your cardiovascular system's capacity for delivering oxygen and nutrients to muscle cells and all cells, and to cart away waste products. So in point 1, angiogenesis occurs in the muscles that you're using frequently in an aerobic capacity. If you're running, your leg muscles, for example, Your leg muscles become better vascularized, meaning new blood vessels grow. There are more blood vessels in the quadriceps of a runner than in the quadriceps of a sedentary person. More blood vessels enables the cardiovascular system to better deliver oxygen and nutrients to that tissue. Within that tissue, if we look at the individual muscle cells, there's a higher density of mitochondria. So if we take a sample of muscle cells from the quadriceps of a runner versus a sedentary person, the runner has many more mitochondria. And during prolonged physical activity, it's the mitochondria that are the major supply of ATP. More mitochondria, more endurance. And then in the heart also undergoes changes that increase the maximum stroke volume possible and increase the maximum heart rate possible. So the heart of a trained athlete has a higher maximum cardiac output. So training dramatically improves cardiac reserve. So a trained person, shown on the right, compared to a sedentary person, represented by that couch potato, has a physically larger heart. This is called hypertrophy. And hypertrophy could be good and hypertrophy could be bad. It also occurs in some disease circumstances. But here, hypertrophy is good. The trained heart is larger, and a larger heart has a higher stroke volume. A larger heart can pump more blood with each beat. And because the trained person has a higher stroke volume, they're going to have a lower resting heart rate. Sometimes they might even have resting bradycardia. So if you have a resting heart rate that's below 50 beats per minute, that's called bradycardia. And in some circumstances, it might indicate a disorder, something worth looking at. But if you're a trained endurance athlete and you have a heart rate below 50, that's not abnormal or anything to be concerned about. So if we think about why a trained endurance athlete exhibits resting bradycardia, this slow resting heart rate, we can think about their cardiac output. If every body, regardless of training, requires a cardiac output of roughly 5 liters per minute, and this person, this trained athlete, has a slower than average resting heart rate, they need to have a larger than average stroke volume in order to maintain an adequate cardiac output of roughly 5 liters per minute. So with this larger heart, they have a slower heart rate that allows for a few more microseconds of time for the ventricles to fill. So the trained athlete, for every heartbeat, they're going to have a greater end diastolic volume. There's going to be a higher amount of blood in the ventricle at the beginning of every ventricular contraction. And so with every ventricular contraction, they're going to have a greater stroke volume. This is also a reminder of the Frank-Starling law of the heart. The greater the end diastolic volume, the greater the stroke volume. This is an animated Wiggers diagram. I love this image. I hope you spend an hour with this image just nerding out over it. There's so much information being presented here, and I hope the way you're viewing this video, I hope you're able to see this as the GIF. This is not a static diagram. It's changing very quickly in kind of an anxiety-inducing way, but it's fascinating. So I hope you spend some time here. We can see all sorts of changes that happen over the course of a single heartbeat. Here are some supplemental videos to help reinforce what we've covered today. And this last image is just a supplemental picture showing you the Frank-Starling law of the heart, which is related to the length-tension relationship. It's just an additional diagram in case it's helpful for you to understand that. So that brings us to the end of cardiovascular system part 1, where we focused on the heart. The next video to watch is cardiovascular system part 2, which focuses on blood vessels. And if you have any questions, I encourage you to post them to the discussion forum in Canvas for everybody to see. And if you're not comfortable doing that, then of course send me a Canvas message and I'm happy to help you out. Thanks for watching.